Compared to organisms like us, bacteria reproduce very, very quickly. So they can go from one cell to two cells in just a matter of hours. This rapid cell division is through a process called binary fission. And so just want to remind you that in eukaryotes, cell division is called mitosis, but this is not mitosis, it's binary fission. It's the same basic idea. The DNA is copied and the cell splits into two, but it doesn't involve a lot of the parts that mitosis has because bacteria don't have multiple chromosomes or linear chromosomes. They don't have microtubules. So a lot of the things that we see happening in eukaryotes is not happening in bacteria. Again, as I mentioned, it can be very, very quick. So while a human cell might take 24 hours to divide, a bacterial cell will only take 1 to 3 hours. And so if you start with a single cell that's invisible to the eye at 9 in the morning, by lunchtime, you can actually see what's called a colony. So one cell goes to a large colony in just a matter of hours. And a significant piece of the idea of a colony is that because it came from one cell and then grew into this colony, they are genetically identical. So a colony should contain cells that are all identical to each other and to the cell you started with. The only reason why they would not be identical is if a mutation had occurred. So these are colonies that you can see on a plate. Again, these become visible to the naked eye after they've divided. So when we're talking about bacteria, because they only reproduce asexually, then parent and offspring are identical. So the only way for us to get genetic diversity in bacteria at first glance is new mutations. So if you get a mutation here, then your offspring are no longer identical. Because bacteria do reproduce so quickly, so again, just a matter of hours, that does cause a relatively rapid rate of spontaneous mutation. So every time you reproduce, there's a chance of a mutation. If you reproduce really fast, there's more chances for mutations. And this allows for relatively rapid evolution. So we have seen over our lifetimes even that bacteria are dramatically different from the bacteria that were around 20 or 30 years ago. And that's where you get problems like resistance to drugs. However, what scientists realized was that even when you take into account how fast they reproduce, they were still evolving even faster than the rate of new mutations. And that's what led them to realize that bacteria are also capable of something unique called genetic exchange, or it's also called horizontal gene transfer, which allows for a related process, horizontal evolution. So horizontal is referring to having one bacteria able to pass its genes to another bacteria that is not its offspring. The traditional route of genetic exchange is from a parent to offspring. And this is what we call vertical evolution or vertical gene transfer. So again, that's kind of the more normal. That's what we see in all living things. Some bacteria and archaea are capable of this horizontal transfer or this genetic exchange to organisms that are not offspring. And so if this bacteria has some mutation, it's able to pass that mutation to organisms that are not its offspring. Again, this is only happening in a limited number of organisms. It does not happen in organisms like us. So these different ways that bacteria can do this, and this is a review, so this is a refresher from your previous biology class. So we have conjugation, transformation, and transduction. Conjugation is when one cell physically interacts with another cell and is able to send DNA over to it through the opening between the two cells. only some cells can do this. Transformation is when one cell dies and another cell is able to pick up the DNA from that dead cell and pick up any possible mutations. And then the third type is transduction, where a virus accidentally transfers a piece of DNA from a cell that it killed to a new cell. And so these are three ways that we can get, again, movement from one cell to a cell that is not its offspring. The impact of this horizontal gene transfer, or this genetic exchange, is huge. So it increases the genetic diversity of bacteria significantly on top of the mutations that they're getting just due to their rapid rate of reproduction. Again, we see it in archaea and bacteria, not in other organisms. And so over the period of that these organisms have been around, this is having large genetic change, or causing large genetic changes in these organisms. So it has a big impact on the evolution of bacteria and archaea. And so looking at E. coli, for example, which is one type of bacteria, at least 17% of its genes, so that's almost a fifth of its genes, came from other bacteria through horizontal gene transfer. And estimates are that at least 80% of prokaryotic genes have been transferred in this way at some point in evolutionary history. So again, this genetic exchange or horizontal gene transfer is affecting the traits of bacteria and archaea quite a bit.